Hi, I'm Phillip McCown, and welcome to my poster, Improved Long Non-Coding RNA Annotation and Detection in Human Transcriptomic Profiles. My colleagues and I are from the laboratory of Dr. Matthias Kretzler, where we conduct research in numerous aspects of kidney disease. One such facet of our research pertains to long non-coding RNAs, or lincRNAs, RNAs that do not typically encode for proteins and whose transcripts are longer than 200 nucleotides, yet are transcribed and often processed or spliced. More lincRNAs are determined to be involved in disease processes and progression in human health. As such, we sought to find as many lincRNAs as we could that are also likely to be present in detectable amounts in RNA sequencing projects. Also, if we identify lincRNAs that are only present in a unique cell type, this may also further eliminate possible functional processes that have not been known before. We sought to find more lincRNAs, especially from resources that might not have been included in ensemble gene annotation. To accomplish this, we combined an ensemble gene transfer file, or GTF, with a GTF from the lincRNA knowledge base, or lincRNA KB. We call this combination the LEG, for lincRNA KB ensemble GTF. After ensuring that the resulting combination did not have any redundant gene entries, we used the LEG in several bulk RNA-seq projects and single-cell RNA sequencing experiments to test robustness of the GTF in several experiments of the same type, and also in different RNA sequencing techniques. To test our GTF in bulk RNA-seq experiments, we obtained several bulk data sets from kidney biopsies from diabetic kidney disease patients that were available in the Gene Expression Omnibus. In the left center, in panel A, are the number of and relative expression of the lincRNAs that are found using the ensemble-only GTF. In panel B are the number of and relative expression of the lincRNAs that were found using LEG from the same data sets that were analyzed in panel A. For both panels A and B, lincRNAs whose expression was above the log2 value of 5 were magnified for their respective panels. It was abundantly clear to us that the LEG found additional lincRNAs that were present in the same data sets, as 3,003 lincRNAs were identified with just the ensemble GTF, while 25,350 lincRNAs were found using LEG. Some of the patients' data sets had indicators of early or advanced diabetic kidney disease, or DKD. These annotations form the basis of a differential gene expression analysis of early versus advanced DKD. In panel C is a volcano plot of the annotated genes and the relative expression values, which show that there are several lincRNAs that are only found in early or advanced DKD, indicating that there are several potential biomarkers of relevance that might be useful in analyzing DKD progression and might also point to several underlying mechanisms in this progression. We wanted to see if LEG could find additional lincRNAs in single-cell RNA sequencing datasets, as seen in the center right panel. Single-cell RNA-seq datasets from kidney biopsies from non-diseased patients were reanalyzed with LEG. Despite the additional lincRNA annotation in these datasets, they were still capable of forming distinct clusters via principal component analyses and Seurat, which could readily be identified as distinct kidney cell types as indicated in panels A and B. Panels C and D indicate that several lincRNAs are present in either a unique cell type or a very limited number of cell types. As with the bulk RNA sequencing analysis, these lincRNAs can be followed up for additional biomarker and functionality testing. Especially for lincRNAs that are only found in one cell type, the knowledge of these lincRNAs only being in one cell type may also be helpful as representatives for specific cell types in other data analyses that cannot resolve transcripts on an individual cellular basis. Together, we found that LEG can identify numerous lincRNAs in both bulk and single-cell RNA sequencing experiments, specifically in kidney biopsies. These lincRNAs could be associated with DKD and with selective kidney and immune cell expression that can be overall informative of a patient's health. Here, we also identified several lincRNAs that were not known to be associated with DKD or immune cell lineages, which may aid in functional elucidation in kidney diseases. Collectively, these robustly expressing lincRNAs could be useful in finding additional and potentially novel disease markers and possible mechanisms for disease progression. Thanks for stopping by, and feel free to ask me any additional questions you might have. My email is pjlmac@umich.edu. I am also attending the ISMB conference in person and can definitely field any additional questions you might have.